director of the Cystic Fibrosis Research Lab at Stanford University, as well as a Benjamin Scott Crocker Professor of Human Biology. His daughter's CF diagnosis in 1981 was the impetus for his interest in CF research. Dr. Wine has received numerous national funding awards throughout his career, including funding from both CFRI's New Horizon and Elizabeth Nash Memorial Fellowship programs, and he has made pivotal discoveries that have dramatically increased our understanding of the disease. Please help me welcome Dr. Wine for his presentation, Fast and Accurate Measures of CFTR Function in Humans.
These are the skilled hands that can dissect submucosal glands,
but they can't plug it in.
They can't. Yeah, that's right.
Which one is it? This one here?
It is, yes. So a couple things. First of all, I was hoping to come up onto the stage the way Isabel did. But I thought better of that. The second thing is I usually really spend a lot of time practicing and trying to get my talk just right. But I thought today I would just stand up here and say whatever came into my mind, no matter how stupid. And who knows, I could be the next president. But the third thing is actually serious. I would like to depart from the way we've done things up to now, because I'm going to be talking about techniques. And it's a technical talk. It's very dull. And to get us through it, I think it would be best if you ask questions as we go along. So if it's inconvenient for you to get to the microphone, if you just shout it out, I'll repeat it, and then we could go along, all right? So that way, if some of you are like me, when you listen to a talk for 15 or 20 minutes, you might have had a question, but then forgotten it by the end. So, okay, so today I'm going to talk about fast and accurate measures of CFTR function in humans. And again, just in case some of you succumb to hypothermia before the talk is over, I wanted to do the most important thing and talk about the many collaborators and supporters I've had. So this is a list of all the people here that have worked on this project with us, and many of them are in this building right now, although I think most of them are upstairs at the research meeting. However, if you look at this slide, you would think, wow, this guy's really got a big lab. But in fact, all these people have already gone off to medical school. Many of the other people are not located in the lab, but are over in Children's Hospital at Stanford. And we actually have one person right now who's doing all the hard, heavy lifting. That's Jian Kim. And Jian, are you out there or are you upstairs still? Okay, she's probably upstairs. Anyway, she's the hard worker. I also want to talk about support from CFRI, from the CF Foundation, and from Vertex. So here's what I think is the good news, and for those of you who may be not so interested in the details of how we do our experiments, this is, I hope, the message that you'll take away. We are in the end game for CF. I missed the very first part of this morning's talk, but perhaps the same point was made. The fundamental science for CF is very firm now. It's a monogenetic disease. It has some modest genetic modifiers. However, the main modifiers are profound environmental modifiers. And as you heard, the story of Cody, told to us by Ginny last night, emphasizes that. So even somebody who is born with a rather mild form of CF variants in their, what would be or should be a mild case of cystic fibrosis, a pancreatic sufficient case, if they get diagnosed very late and therefore treated very late, the consequences of severe. On the other hand, we have one, and sadly only one, set of parents for a newborn. If you have a newborn child with CF and you treat that child, as we have advised over the years, aggressively, they will end up living a full, active, and healthy life. We know the gene and the gene product, they're understood in increasing precision and depth. And most importantly, and this is why you want to stay healthy with your treatments, systemic drugs, pills that precisely target the basic defect are now in the clinic. More effective ones, much more effective ones are on the way. And these will treat more than 90% of the people with CF. And this was Dr. Dinitz's talk this morning. Large efforts are underway seeking cures for non-responsive mutations and for ways around them, as he discussed with the sodium channel blocker. Okay, this is the focus, and I want to make that system, I want to make the search for these systematic drugs go much faster. How do we get these better drugs faster? Clinical trials are slow and expensive, and we're running out of patients. Patients are in so many different trials now that it becomes laborious to get new ones. But there's an alternative, the N-of-1 trials, such as those that have been conducted by Dr. Nielsen, where each subject serves as their own control. And you do these trials so that you're directly affecting, you're measuring CFTR function right where it's happening, not some downstream function like lung function, which might take years to develop or change. You quantify effects for a single subject in a way that provides you a lot of confidence in the results, and you use multiple parallel measures for each subject. So how do you do all this? Well, it turns out that the sweat gland function is well known, and it's a perfect CFTR assay. It has extreme advantages that have been capitalized over the years, in particular by Paul Quinton. The sweat gland is an unusually accessible organ. Each of us has over 2 million sweat glands covering the surface of our body. These are not affected by infection or inflammation in CF. And the sweat duct, as you know, has a CFTR-dependent salt absorption mechanism. That's the sweat test. And Marty Karazi has recently written up a nice little discussion of the sweat test in Lancet. Today I'm going to talk about a different part of the sweat gland, and that is the sweat coil, this part down here that actually produces the sweat. And it does that in two different ways, and that's the secret. One way requires CFTR, and the other way doesn't require CFTR. So we can compare these two and learn a lot about CFTR function. So this is the basic idea shown here. This is called the bubble assay, because we're looking at bubbles of sweat that form in an oil layer on the surface of the skin. This would be the schematized sweat glands stretched out. And these are the two pathways that make the sweat occur. Normally, when we sweat, both of these are active. This way, the cholinergic way, uses a pathway that doesn't require CFTR, and this one does. We call this one M sweat, and this one C sweat. So here is the apparatus that we use to study it, and another apparatus that you're going to hear more about tomorrow called the evaporimeter that can study sweating in another way. So the evaporimeter, which is a commercially available instrument and has many advantages, especially when working with little children, produces this kind of a trace here, and that trace is actually showing you the rate of sweat secretion. You'll hear more tomorrow. And this is the other technique that we use, and it is more, as you can see, more elaborate, but it has a variety of advantages that I will tell you about today. You're going to hear more about this from the evaporimeter from Dr. Salinas tomorrow, and now I'm going to tell you about how the individual sweat gland measurement technique works. So if you look through the camera down onto the surface of a human being who is sweating, this shows you the result of a stimulation of M sweat. This is the kind that works in people with and without CF, and you can see these individual sweat bubbles. Now, the amazing thing, and what makes this a methodology so powerful, is that we can go in there and label these. We can give them a number. That's gland number 1, number 2, number 3. Then we can come back after cleaning off the arm and putting oil on and stimulating with a way that only works in cystic fibrosis, the CFTR dependent method, and we see those same glands. They're in the same position, and they stay there forever, as far as we know. So we can test that same gland for weeks and for months on and off drugs. So some of you were in the research session and saw these human lungs that AK was working on, and the tremendous difficulty in getting lungs after transplant to do some physiological work. We did that kind of work for over 10 years, and in addition to getting up in the middle of the night, the sad thing was that the moment you started to work on the tissue, it was dying. And when you were finished with it, you could never, ever go back and study it again. Whereas this subject walks into your lab and says hello to you, and then they walk out, and then they come back, and then they come back again, and so on. And many of you out here, I can see some of you have actually done that, and we're very grateful for it. But you can see the advantage of having a living human being. The other advantage, of course, when you're studying drugs, it's crucial to know how drugs work in a dish. In fact, you must first prove that they work in a dish before you could ever think of putting them in a human. But once you go into humans, all kinds of strange things happen with the drugs. They get bound up in the blood, and they get filtered in the kidneys and so on. And this takes that all into consideration and gives you a final output. So if we take this sweat bubble, the M rate, and this sweat bubble, the C sweat rate, we get a ratio, and that ratio turns out to be an exquisitely sensitive and accurate measure of CFTR function. So I told you this was a technical talk, and so I'm focusing in on one kind of technique, but I want to give you an overview of how it fits into our entire research project. But now let me stop and see any questions to this point. Who doesn't understand something I said?
Is sea sweat always blue?
Ah, good question. Before we give that test, we make the person drink a great big gallon of, no, just kidding. That's a great question, thank you. Sweat is always clear. But when people have cystic fibrosis and are taking a drug, we will see later, they produce a very small amount of sweat. And the sweat bubbles are so tiny that we wanted to be able to have a method that we could be sure we could see them no matter how small they were. So what we devised was that the oil is filled with little tiny particles of water-soluble blue dye. But because they're water-soluble in the oil, they're just particles, so you can't see them. But as soon as they partition into the aqueous phase of the sweat bubble, they turn the sweat bubble blue. So great question, thank you. That's the kind of question that we need so I'm not leaving everybody high and dry. Yes, Daniela.
How do you go back to the same spot if you're testing the same subject?
Yes, so that's also a great question. You can't really see it very well here, but almost everybody has some kind of freckle or imperfection on their skin that we use as a landmark, and that's crucial. We have had 2 subjects where we couldn't find anything, and so they did their best to try to mark it, and it didn't work. And in those subjects, we couldn't go back to the same spot. But once we have that spot marked, we now have a very good way of getting back to it, and that leads me to this point here. So this is what we call our sweat gland program, and it has 3 big components: system development, sweat gland biology, and drug discovery. And all of these arrows just mean that all of these things are mutually reinforcing. So if you make a better methodology, then you can learn more about the biology, and the more that you know about the biology, the more it helps you do the drug discovery, and so on. So for system development, we have an improved positioning system, so we can come back within 1 millimeter to where we were, or less than 1 millimeter. And we want to improve the imaging. We want to have automated scoring and a variety of other things that you don't need to know about. But we're working on the system itself to make it better. And the goal of that is to really understand sweat gland biology even better than we now do. And we're going to do a couple of things that I'm going to talk about in detail today. One is CFTR function in healthy control subjects, and that's where some of you have helped us today. And we're comparing sweat rate versus the standard sweat chloride method that is the gold standard. And we're working on ways, and this is really tricky, and others that are working with babies, for example, you'll hear about tomorrow. How do we optimize the way that we stimulate? So now we have to inject these drugs with a small insulin needle underneath the skin. And finally, all of this is to be at the service of drug discovery. And so far, Vertex is driving this because Vertex has the only systemically acting drugs that directly improve CFTR function. And ivacaftor you've heard about with various kinds of mutations, or Combi is coming along. And then these are the drugs that have me euphoric with excitement as they come online and will be, I think, really putting us into the end game of CF. And so we hope to be a part of all of this. But today, I'm going to focus on CFTR function in healthy controls and just tell you why we're doing that, why it's important to do. Okay, so CFTR function in healthy controls. How do we do that? Well, let's say this is healthy control.
You said,
yes.
You said to interrupt you. So
yes, please do. Absolutely. Thank you, Doug.
Okay, so here's a really big picture question that occurred to me while I was listening to you. You started off saying, okay, the sweat gland is this elegant organ, it's preserved, we can go back and access it again and again. All those things are great. How do you know that if you fix the sweat gland, you're going to have any effect on the lung? Or on somebody's, condition of having CF?
That's great question. So did everybody hear the question? How do we know that the sweat gland has anything to do with anything else? The reason that we know that is because cystic fibrosis is a monogenetic disease. So the CFTR that is in the sweat gland is exactly the same CFTR that is in the lung. However, because Doug is smart, he's anticipating that there's different ways that CFTR can function. So CFTR, for example, is working in an absorptive function in the sweat duct. That's what Paul has studied for such a long time. And in certain situations, it seems to have more to do with bicarbonate secretion than with chloride secretion. Nevertheless, theory tells us that if you make CFTR work better anywhere, then it's going to work better. The functions will be improved and perhaps be improved even more, maybe even amplified. That's a theoretical answer. The experimental answer is that in those few cases now, we don't have many, where you've looked at what the sweat gland is doing and what the person is doing, they correlate well. So there was a very good big change in the sweat duct function with ivacaftor, and I will show you later on what happened with the sweat secretion with ivacaftor. And there's another question. Yes?
Were your healthy control subjects both carriers and people without any CFTR?
So today, the question was, were carriers included in this control group? And I'm going to focus today on just healthy non-carriers, but we're also looking at carriers as well, all right? But we decided to actually separate them out. We started with them together. We're now separating out the carriers, but eventually we want to do a whole big slew of carriers. And there's a technical reason for why we're doing that. Okay, so what does it mean to, so I got discombobulated there. So what does it mean to, for CFTR function, what does that mean? Well, if we bring in a healthy control subject, number one, we measure their M sweat, their CFTR non-dependent sweating, in about 50 glands, C sweat in about 50 glands, and we do this 3 times, take the average for each gland, and then we get 50 average CM ratios, which gives us a subject mean. To find out how good we are in injection and so on, we look at C1, C2, C3 consistency, M1, M2, M3 consistency. I'll show you what I mean by that. We also then correlate these results with sweat chloride results, and then we finally do a nasal brushing to look at CFTR mRNA, but that's not ready to talk about yet, unfortunately. And we do this 50 times for 50 different subjects. So why is it important to quantify sweat rates in healthy controls? And to answer that, I need to examine how this is related to CFTR function. And here is, again, if you're going to take one thing away technically about this talk, this would be the slide to take away. It's a complicated slide, so let me run through it. So this is the hypothetical CFTR function going from 0 to 100%. And this was established by our method, actually. And so one of the things we want to do is validate it. And what you see on the sides here are 2 different ways of assessing this CFTR function. One is the sweat chloride method, and one is the sweat bubble CM ratio. And this is now expressed as a % of the wild type average. Based on old work that we did, there's a linear relationship between the CM ratio, the ratio of CFTR dependent to non-dependent sweating, expressed as a % of wild type, and the amount of CFTR function. How do we know that? We know it, unfortunately, because we only have 2 big points on this graph. One point is CF, they don't have any function, 0. And the other is carriers. They have 50% of function. That's really cool. And this, when we did this, Paul Quinton and I and Julie Desch, where she is, yes, who was the first author on the paper, when we were all much younger, it was really the first time that anybody had shown that carriers had anything that could be shown expressed in this way in a believable way. So let's contrast that with what happens with sweat chloride. Now, if you look at sweat chloride in carriers and compare it to wild type, you can barely tell the difference. They're almost exactly the same. Why is that? It's because there's a logarithmic relationship of sweat chloride to CFTR function. Really interesting that that is the case. And what that logarithmic ratio means is that sweat chloride is an exquisitely sensitive thermometer, if you will, for tiny changes for CFTR away from 0. So if you have no CFTR, your sweat chloride is going to be up in the hundreds range. Add a little tiny bit of function, and it drops like a stone. So with pancreatic sufficient subjects, it's under 80, a big drop. And now you add an ivacaftor, and it'll drop down here in various tests to almost 40. And remember, then what happens as you go out along here, as you start to get better and better and better, with, we hope, better and better drugs, it's going to be harder and harder and harder using the sweat chloride method to tell the advantages of these improved drugs. But this assay will give you a nice readout. Okay? Any questions about this? Or, I know it's complicated, but yes, there comes one.
Jeff, actually, a very quick question because my daughter has been on Orkambi for a year.
Yes?
And they did the sweat chloride test before, they did it, after the protocol. And basically what happened is, I'm so sorry. Basically what happened is that her starting sweat chloride was 101. The end test was 99, statistically insignificant.
Yes?
So basically, does it mean that Orkambi doesn't really work?
All right, I'm going to go out on a limb here based on a very small amount of work that we have done with Orkambi and also some of the things that are in the literature. I think Orkambi, for reasons that are unclear, I think, to everybody, Orkambi has quite, let me change it back. I was going to say Orkambi has variable effects. I don't believe that. Orkambi always does the same thing. It's the people that are different. So some people with CF respond well to Orkambi, very well. If that weren't true, it could not possibly have been FDA approved. But some people just don't. And one possible way to kind of test that out is the sweat test. Okay? Another way is to wait and see, again, if you talk to Paul Quinton, maybe there's an effect on bicarbonate secretion somewhere that's not going to show up in the sweat test at all. I guess we're coming full circle back to Doug Maudlin's question. So if the sweat test gets better, everything's going to get better. You can be real happy. But if it doesn't get better, it's not definitive proof that you won't see effects somewhere else. Any other good effects that you can see? Any weight gain, for example?
Actually, she has been very stable.
Okay, I'll take stable. I'll take stable. All right.
So Jeff, I have a question for you too.
Yes?
Over here.
Oh yeah, Marty.
What can you tell us about age effects? Sweat chloride tends to go up with age, right? Does this I mean, I ask that because I'm just curious if you have much with. I mean, how long have you guys been doing this? Not so long, but have you looked at it with age to tell us whether there is going to be a change in the ratio?
All I can tell you is we've looked at, no, is the short answer. But we see, all I can tell you is some really old people sweat okay, and some young ones sweat actually less, have the CM ratio. And we think for our test that the absolutely most important thing is the number of CFTR channels. I'll come back to that. Now, in terms of why the sweat chloride changes with age, we have some experts in the room, and I would defer to them. My understanding, but Paul can correct me, my understanding is it probably has to do with the rate of sweat secretion more than anything else. But Paul, is that right?
That's basically what I was, all the other factors involved. In fact, there are a lot of factors involved. But as you grow and get older, the more the sweat better. Sweat glands are a little bit like muscles. You get better when you
Yeah.
get older. Then it's complicated because the amount of sweat that gets absorbed. As the sweat gland gets better, able to secrete more, If it has the same amount, same ability to reabsorb, the fraction that's reabsorbed is less. So the concentration goes up as the sweat rate goes up. As you get older, I think it stays more or less the same, but I have seen perfectly normal people sweat 80 millimolar chloride, and I'm sure they didn't have CF, but they sweated like hell. So really high rates will give you higher chloride concentrations. Now, that's a simplistic answer, but an answer.
All right, great. Yeah, sure, absolutely.
Since it's related, I'm sorry I'm being stubborn. This comes to this second part comes to bear because of a recent paper actually that came out also by Daniele and a few of us on the 5T, TG 12 and 13,
Right.
which is showing changes. So someone's sweat is low or like in the 20s, and then over the next few years we're up to 60. What's happening there? Will this ratio be able to tell us what's going to happen for this person, or will it change too?
I think one of the talks said, I think that my best answer is, I don't know. Let's talk. I think that would be a good thing for us to talk about offline, because I think it is going to be a pretty complicated answer. Really interesting issue, though. Is that okay? Is that, I invite questions and then I say, I don't know. I could be president. All right. So here's why it's important to do this work in healthy controls. I told you it gave you a linear readout, and for every scale, so let me tell you, so the geeks in the audience will know that if you have a measurement system with a true zero, that's the most, there's a scale of measurements. You can have nominal, ordinal, interval, or ratio scales. If you've got a true zero, you can have a ratio scale, the most powerful kind of metric. That's really good. But you also would like to know if there's an upper end to it, right? So, and in our case, what we're doing is to make sense of this, since just a CM ratio of 0.16, what does that mean? The only way to express it, I think, in a meaningful way is to express it as a percent of wild type function, which means you better know what wild type function is. And wild type function, now that we can measure it precisely, turns out to have a broad range. That's another take-home message. Like a lot of other things in your body, when you go to the doctor and they take blood tests and they show you have this much of this and this much of that, and there's a big range, and you're sort of at the low end, and you say, oh, I'm at the low end. And I say, it's okay, you're in the normal range. And now we can do that with CFTR function. And why is there a range? Well, because CFTR channel number changes per cell, CFTR channel function changes in disease states. Other factors might affect CFTR, and changes in CFTR can be produced by drugs. And we can now parcel out, we're going to try to parcel out these differences in living human subjects. So that's really kind of nice. So this is what I just told you, the rigmarole we go through to find out what CFTR function looks like in a healthy control. Well, what do the data actually look like? I'm not going to show you all the data, but I am going to show you the compressed data. And let me walk you through this slide. So what you're seeing along here is the rate of M sweat in nanoliters per minute. And what you're seeing along here is the rate of C sweat in nanoliters per minute under the conditions of our testing. So they have to be carefully defined. Now, what you're seeing here, each one of these dots represents 6 tests, 3 M tests and 3 C tests for that single gland in the subject, okay? So this is a degree of precision that's hard to come by, but I think if you're going to do precision medicine, maybe you need to have precise measuring metrics. So we tried to do that. So why are these glands spread out all along this rate? Well, it's just what Paul Quinton said. Your glands have different sizes, and they get bigger when they're exercised. But they seem to be, some of them, intrinsically bigger. And so when you were microperfusing ducts, I think you saw little ducts and you saw big ducts. And so there are big glands and there are little glands, and that seems to be the major thing that determines the volume of sweat produced when you stimulate with the mechanism that doesn't require CFTR. What about the mechanism that does require CFTR? Well, you can see this follows along really nicely. So for these 52 glands, there's a correlation coefficient of 0.90. That's really quite good. Well, okay, then what about, so these ranges here are variations that we think primarily are reflecting our experimental conditions. So we think if we could do everything perfectly, everything perfectly, these would shrink to much smaller dimensions. Given all of that, look here, you see, let's take this guy. He's the farthest above the line. That's the highest M and C sweat rate, the lowest C sweat rate, big range in these glands. We can plot these data in a different way. We can say, okay, because these are so well correlated, we can do the C-M ratio and ask, how does that vary as a function of the M sweat rate, the basic sweat rate, okay? And that's what's shown on the next slide. So now this is really interesting. Now, the correlation coefficient between M sweat and the CM ratio, now it turns out to be 0.4, non-significantly. It's basically a flat line. It's not significantly different from 0. So that's exactly what we wanted to do with this ratio method. We wanted to make it irrelevant how fast the gland was sweating. But scattered along this essentially straight line, look at this thing here. Here's a gland up here, and here's a gland on here. Now we're measuring the ratios. So these glands are pretty much the same size based on their M sweat rate, but this one has about 1/5 or 20% of the CFTR dependent function as this one. Why should that be? Well, the first thing that might jump into your mind is, by the way, this is just a distribution of all these responses. The first thing that might jump into your mind is, well, there's big error bars. Maybe this is just an experimental artifact. Maybe we mismeasured it or something. But no, we can go back and look at this particular gland. This is gland number 42, and in this subject, here's that gland. So here's focusing in on 5 sweat glands, and this is gland G42, 10 minutes of M sweat. And here's those same glands after the person's drank this big bottle of blue dye. There's G42. Now, if we circle all of these glands here with these little yellow line and circle them here, you can see that even though this went for 30 minutes, these are all smaller, and that's what we expect. C-sweat is slower, but not this one. This one's actually larger. So for some reason in this, what we think, if we could do the experiment, what we think is if we could dissect out that single gland and stain it, we would see that it's rich in CFTR. We're not sure why that should be, but people who have stained in the lung, for example, find certain cells that are just richly expressing CFTR. So it's a biological outlier is what we're calling it. The point is, we know about it, we can take it into consideration, we can think about what it might mean further down the line. So what accounts for CM variation within a subject? Well, all of these things vary. We can rule out gland size by taking the ratio, and then we're left with all of these things. And finally, we have experimental error for both of them, and that's what we are concerned that we minimize as best we can. That's why we're developing the systems. These are cross-test correlations. All of these are for one subject. So let me explain what this means. So this was the first test on this subject, stretched along this line, and the test number 2 for this one. So this dot here, for example, this gland produced 4 mLs of M sweat on this test, and 2 mLs on test 2. Now, that's not a very good agreement. But overall, the agreement's not bad. If they were perfectly aligned, they would align along this red dotted line. And so you can see the scatter here. And that same thing goes through all of these for M and C sweat. And you can see that the ratios here, the correlation coefficients go from, I think the highest is 0.91 and the lowest is point something. Oh, 9.96 down here is the best. Anyway, so this is what we do to check ourselves. Why do we do this? Well, sometimes we will see that the correlation coefficient is 0. And when we go back and say, why is that? We find out it's because we were at the wrong spot. So this is a cross-validation mechanism that helps make this a powerful way to proceed. Okay, now all of that was for a single subject. Now what's the range of CFTR function among healthy subjects? Now if this is what you see for one subject, what do you see if you take these kind of data and compress them all down into one single average point along both of these lines? And our hypotheses based on prior data is that healthy humans differ in their level of CFTR function, that we can detect it, but that they are consistent over time for an individual. So I can't really answer Marty's question about age, but over weeks or months, they don't change. And we think that the variation within controls bears directly on assessment of CFTR-directed therapies. And so I already told you the methods, and so we can look to the data. This looks a lot like the graph that I showed you before. Except now, instead of looking at individual glands, you're looking at individuals, individual people. These are all healthy controls. And now, what you see here for that dot represents 50 glands. They were each tested 3 times with 2 different tests, and all that data coalesced and compressed to give you this data point. And so now what we see across individuals is people who have tremendous variation in the amount of CFTR function. These are normal, healthy individuals. And these people have CFTR, well, I didn't know. So why is it spread along here? I'm not showing it on this graph, but if we broke this down by gender, you would tend to find males over here and females down here. However, there's some very big exceptions to that rule. If you have somebody who works out a lot, if you have a runner, a woman who runs a lot, she's going to be moved over here to the right, okay? It's because she's exercising her sweat glands and she moves into the place where men typically are. But now if you have men who are very sedentary, they're going to be down at this end because of their low M sweat rate. So again, that's why we want to take a ratio and find out what's the ratio look like. So now, instead of plotting C sweat versus M sweat rates, we plot this C sweat, M sweat ratio rate versus time. And now this is a flat line. So now all of these are the individuals. And what this is telling us is that regardless of how fast a person's sweat glands are producing sweat when they're thermally activated, when the person's running or something, regardless of that, there's this huge range in their CFTR-dependent sweat rates. So this person up here, for example, this subject has the highest CM ratio that we have discovered so far. Now, if you think about this, we're expressing this as a percent of wild type. There's a little bit of circularity here, right? How do we, you know? So what we're doing is taking we're setting the average of all these relative to 100%. So this person, who might actually be in the audience, is champion CFTR producer, is what I would argue. That means that on average, this person has about twice as much CFTR function as the average individual. And this person down here has such a low level that we think that these subjects down here, when we bring them in and have them genotyped, these were not genotyped. When they are genotyped, these are in the range of heterozygotes, and some of them might well be heterozygotes. Some of them also might have, well, I won't get into all the details. But anyway, you can see this tremendous range.
But truly that balanced out to a horizontal. Is that what you're saying there?
Yeah, so this is, you know, this isn't me, this is Excel doing this, and so I didn't cheat. So that's when you ask it to tell you what these points are giving you, that's what it gives you, okay? Any other questions on this? Because this is kind of the main point. We're not going to get through this, by the way, but it doesn't matter. It just doesn't matter. It's just fine to have this kind of back and forth, much more important. Okay? All right. So what about sweat rates and sweat chloride? It turns out that in control subjects, they don't correlate worth a damn. So these are very low correlations between sweat chloride here and the CM ratio. And there's a little hint here that when the M sweat rate goes up, the sweat chloride goes up a little bit. I think if we had done more, maybe it would have been significant, but it wasn't. So the last part of this is, what does C-sweat look like in CF subjects? And I didn't do much of that today, just enough to give you a taste, a feel for where this is all going. So this is the same data you've seen before, it's just compressed now. This is now for a single subject. These are all individual sweat glands for that single subject. Same one, I just kept it to one subject to not confuse us. And this is now a G551D subject on ivacaftor. And this is now the same scale for C-sweat, M-sweat. Notice that this subject actually sweats faster normally for M-sweat than this subject, but that's the C-sweat rate. If I now now here's the same data, but now the scale over here is multiplied by 10-fold. So instead of 1.4, this is 0.14. And now, even under these conditions, you can see that half of the sweat glands in our assay didn't respond at all. They produced no visible sweat, and the rest produced the amount that you can see here, very small amounts. Okay, this is the best subject we had on ivacaftor. Now, how is it possible to reconcile this small response with the absolutely, you heard about it this morning, and some of you know about it from firsthand, the absolutely fantastically good clinical response that you see with ivacaftor? There are many explanations, but the one that we favor is that it just does not take very much CFTR function to really make you a lot better, okay? And so what does this look like? What do these data look like now when, instead of plotting them C versus M, we go back to the ratio method? We plot the ratio versus the rate. Now we see a really interesting and very informative surprise. So this is for that single subject, and you can see here, this is basically not significantly different from a flat line. That's what we expect to see. But what's going on here? This is not a flat line. It just, we didn't know what to make of this. So now, the faster you have M sweat rate, you have a whole bunch of C sweat rate. And the ones that are secreting slowly, we see nothing at all. And that told us that there is a problem with the method. And the problem with the method is, and I talked about this in an earlier talk, there is a subtractive process, or processes. Some of the sweat, that is, we think every single gland in this subject is producing sweat, every single one. But they're producing it at such a low rate that there are subtractive processes, absorption in the duct and the time it takes to fill up the duct and a few other things, that if they're at a very low C sweat rate, it does not exceed the subtraction rate, and so they appear to be zero. And you can show arithmetically that if you have this kind of a picture here, if you have this kind of picture here, and you subtract a constant factor from all of them, you will get this kind of picture here for the ratio. And if that went over everybody's head, it's okay. Just the take-home message is we don't see all of the sweat, so that the actual function here is somewhat larger than you would see from these raw data. And in our published papers, we corrected for that. Even after the correction, you don't have much CFTR function. And I think, am I over time? No. Just about there. Okay, just about there. Okay. Oh, good, because here we are. So the summary is that the CM ratio is linear, and it complements this sodium chloride method. So nobody in their right mind would want to get rid of sweat chloride measurements. They're spectacular. They've really been useful for us. But when you put the two of them together, you really, really get a lot of bang for your buck. It's a very powerful thing to do, and that's what we're doing. And measuring individual glands provides many advantages. So you'll hear, there are ways where you don't look at the individual glands. But if you do look at them, then you get multiple semi-independent measures. You can estimate and compensate for sweat losses. You cannot possibly do that unless you know the rate for each individual gland. You can do paired statistics, and by that I mean you just line up all 50 glands and say, what did those 50 glands look like before ivacaftor? What did they look like after ivacaftor? And you get p-values like to the 0 to the 27th. I mean, they just knock your socks off. They're just astronomical. You get multiple methods for error checking. I just showed you one. And because of all of these things, this kind of assay is really powerful and useful for N of 1 studies. Because when you're doing N of 1 studies, you only have one subject. And so you want to get all the information you possibly can from that one subject. What are our research priorities? Just we want to expand the cohort and correlate with transcript sequence data. We want to set up assay systems at independent sites, and I'm happy to say that we now have them. There's one operational in Verona, there's one operational in Brussels, and there's one getting set up in Paris. And I need to visit these sites routinely. We want to simplify the apparatus and the drug delivery methods. We want to automate the scoring, because this scoring that you have seen was laboriously done by hand. And a special plea, if any of you know anyone who is about to start Orkambi but hasn't, or is going to be enrolled in a VX661 trial but hasn't started, we'd love to see them. And then when they go into the trial, we can follow them. This was done with ivacaftor. It's sort of a side light, and my colleagues in Stanford are very helpful, but there's a lot of people that aren't at Stanford who might know about this, who might also be eligible for these things. And finally, I'm going to end up, we need legislation for drug pricing. But I'm not talking about ivacaftor. Ivacaftor, that's a real drug. A lot of hard work went into it, and it does a real thing. I'm not talking about that. I'm talking about these guys who do nothing except steal. So the drugs that we inject to produce a C-sweat are generic. They were invented 50 years ago. They were dirt cheap. Well, not dirt cheap, but we used to pay, I can't read this something. Can anybody read this? It was anyway, you forget the numbers. Look at the line. So the expenses for our drugs just skyrocketed. So we got CF foundations on the line. They said, "What's with your budget?" And we said, "Well, we have to." So this guy, I hope, is going to jail, and I don't know what's going to happen to him. But this is really bad. This gives drug companies a bad name. Good drug companies get a bad name from bad drug companies like these. These aren't even drug companies. I don't know what they are. They're just crooks. I mean, they're stealing a lot of money. That's all of that from here to here is my definition of theft. This is nothing, no value was added. Sorry, am I venting a bit here? Okay, and I just want to end up with the collaborators. Okay. And we got the questions, right? Wait, okay.
All right, thank you, Dr. Wine. Well, this concludes our very full day of fascinating presentations from a diverse group of CF experts. I thank everyone for their attention and participation. Now, in just a few minutes, we're going to have 15 minutes to stretch our legs and enjoy some